go ahead and get started. So the next speaker is Greg Lang.
Thanks, Mike.
So I'm going to tell you about a project that we've been working on in my lab for a couple of years now, trying to develop a new method for measuring fitness and epistasis among mutations that arose in the context of experimental evolution. So previously, we evolved about 600 populations of haploid yeast for 1,000 generations in rich glucose medium just by serial transfer in 96 well plates. For 40 of these populations, we performed whole genome, whole population time course sequencing to get a pretty detailed picture of the dynamics of adaptation. So I'll show you one example. So this is a single population. Up top, you can see the frequency of all 23 mutations that were in this population. On the bottom is the same data plotted as a molar diagram, so you can see which mutations are occurring on which background. One of the most obvious things that popped out of this data is that mutations don't move through populations as individuals. Rather, they tend to travel in groups that we've called cohorts of mutations that track very tightly with one another over time. So this is one of the examples. The other 39 populations show very similar types of dynamics. The major questions that came out of this are, one, what are the mutations that are actually driving adaptation? Second question is, what are the fitness effects of those mutations? And third, to what extent do epistatic interactions drive adaptation? So the gold standard method for measuring epistasis amongst the various mutations that occurred along an evolutionary path is to reconstruct all possible genotypes of evolved mutations. While this is useful for small numbers, this is difficult to scale up because the number of possible genotypes increases exponentially with the number of mutations. And so this has really limited this type of analysis to at most 5 evolved mutations. So to overcome this limitation, we're taking powerful methods that have been developed for QTL analysis, which you've heard about, and we're applying them to fitness in our evolved populations. So the first thing we do is we make these bulk pools by crossing evolved and ancestral strains. We do this in a slightly different way than most people in the QTL field. So we'll take our MAT-A evolved strain, we'll cross it to a MAT-alpha version of the ancestral strain to create a diploid that's heterozygous for all of the evolved mutations. And we set this cross up so that the evolved strain is also, or that our diploid strain is also heterozygous for the dominant cannabinoid sensitivity gene CAN1 at 2 unlinked loci. We then take this diploid, we convert it to a MAT-AA diploid by forcing a gene conversion event at the mating type locus. We sporulate this diploid by complementing the MAT-alpha 2 gene on a plasmid, and we select for haploids by plating on cannabinoid medium. And so these pools of segregants contain about 10 to the 5th cells. They're in a genetic background that's almost identical to the one that was used in the evolution experiment, but they each will have a random combination of the evolved mutations. Once we have these pools, we do 2 things with them. The first thing we do is we just put this pool in one well of a 96 well plate, and we propagate it forward exactly as we did during the evolution experiment. And we track the frequencies of all of these mutations by doing next-gen sequencing every 20 generations. What this gives us is the fitness effect of all of these mutations averaged across all possible backgrounds in which those mutations could occur. The second thing we do is we go into this pool and we pick 192 individuals, and without knowing their genotypes, we measure their fitness using our standard flow cytometry-based fitness assay. What this gives us is the segregation of fitness within this cross. Okay, so how does this work? So here's a pretty simple population. By generation 545, a cohort of 2 mutations, ELO1 and GAS1, have fixed, and the third mutation in sterol 12 is about to fix. So we took a clone for where you see this red line, so at generation 545, we crossed it to the ancestor, we made our large pool of segregants, and we did the bulk segregant fitness assay. And from this, you can see that 2 of the mutations, sterol 12 and GAS1, have a fitness benefit of just above and just under 3%, whereas ELO1 contributes little, if anything, to fitness. When we look at how fitness segregates within this cross, we get a distribution that has 3 major modes to it. And given what we know on the left, you can sort of interpret that that bottom mode are individuals that have neither GAS1 or sterol 12. That middle larger mode is either GAS1 or sterol 12. And then the largest fitness ones close to the evolved are ones that have both gas1 and sterol12. So this is a pretty simple population. We can do this with more complex populations now. So from this population, we isolated a clone from generation 910. It contained 14 evolved mutations. We did the same thing. We constructed our pools and did the bulk segregant fitness assay. What you can see from this, most of the lines are flat. So most alleles did not enrich in our pool, which means that very few of these mutations are actually contributing to fitness in this population. When we look at how fitness segregates within this cross, we see a similar thing. We see multiple modes, which suggests that the fitness is a low complexity trait here, and there are very few things contributing greatly to it. What I want to do is take the plot on the left and just highlight the largest fitness effect mutations that we see. They're ROT2, IRA1, and MID2. And if we map them back onto the dynamics, you can see that you have one major driver mutation in each of these selective sweeps, and then the other 11 mutations are largely hitchhikers. We've done the same thing now with 12 populations, and so we can start saying something meaningful now in our data about the distribution of fitness effects of mutations that arose in the context of evolution, or experimental evolution. So this is 111 mutations, and their average fitness effect just sort of lined up from lowest to highest, but broken down based upon whether they were caused a change to the amino acid, whether they were synonymous, or if they were intergenic. So from these 111, if we just look at the 65 that were either nonsense or frameshift mutations, 24 of them have fitness advantages ranging from 1 to 9%. Across these populations, we measured the fitness effect of 4 different IRA1 mutations, averaged across the backgrounds in which they arose. And so these are 4 different mutations, looked at across 4 different backgrounds, and they cluster pretty nicely. That's not always the case. We also had 2 different KEL1 mutations that we looked at. So these are 2 different missense mutations that occurred in 2 different populations. They have very different fitness effects. There are a number of things that could explain this. As I'll show you in a few slides, we actually think it's because in one population, KEL1 is acting epistatically with other mutations. I should note that we also have one and only one possible deleterious mutation that fixed across these 12 populations. And we also have one interesting possible beneficial intergenic mutation. So how do we determine now if there are epistatic interactions among the mutations in our populations? So that's sort of the key challenge, and I guess why this session is here. To try to get at epistasis from our bulk fitness assays, we're extending a framework that was published by Dan Weinrich a few years ago, and so I want to sort of go over what this does. And so what Dan showed in this paper is it's possible to convert between, since we're not having a pointer is going to be tough, but it's possible to convert between the full fitness landscape or the full genotype to phenotype map shown here in W to an epistasis matrix that pulls out the epistasis terms. So this W matrix, I'm showing the full genotype to phenotype landscape for 3 mutations. So at the top is the fitness of the ancestors, so no mutations. You see the fitness of the singles and all the way down to the fitness of the triple. So if we calculate, so this is called the Hadamard transform, and if we calculate the first term in the epistasis matrix, you can see that it's based on the expression for the average fitness of all possible genotypes. If we do it for the second term, rearrange the terms, you can see that it's now the fitness of mutation A on the ancestral background, the fitness of mutation A on the background of B, A on the background of C, and the fitness of A on the background of BC. And so this is an expression for the fitness effect of mutation A averaged across all possible backgrounds on which it could arise. I'm going to show you this 4th term. So if we multiply through, rearrange the terms, you can see that this is an expression for the fitness effect of putting A on the background of B minus the fitness effect of putting A on the background of the ancestor plus the fitness effect of A on the background of BC minus the fitness effect of putting A on the background of C. So this is an expression for the average epistatic interaction between mutations A and B. And you can fill out the rest of this matrix, including the higher order epistasis terms, in which in this case there's only one. What we want to do is we want to invert this logic. We want to start with an epistasis matrix and see if we can predict the full genotype to phenotype map. I already told you that we can measure the fitness effect of all mutations averaged across all backgrounds on which they could arise. so we can fill in some of these values. We can then calculate the average fitness of all possible genotypes, which in the case of either no epistasis or generic diminishing returns epistasis, which you just heard about, it's just the sum of all the average fitness effects. And then we can start plugging in values for epistasis. The simplest thing we can do is just set all of the epistasis parameters to 0. And we can ask, can we predict the full genotype to phenotype map? Can we predict how fitness should segregate in this cross without invoking epistasis? And we can go back in and do something similar. We can now substitute in, rather than no epistasis, just generic diminishing returns epistasis, what Jose talked about, basically just a progressive tax of putting mutations together. So for most of our populations, we can indeed predict how fitness segregates in the cross, invoking only generic diminishing returns epistasis. But not always is that the case. Here's a population that looks pretty simple, actually. You had one mutation that was sweeping through the population. It got outcompeted by this cohort of 11 mutations. If you look at what those 11 mutations are, you notice that 4 of these mutations cause changes to the protein coding sequence of genes whose proteins localize to the bud neck or sites of polarized growth. And so there's a first indication that maybe there's epistatic interactions happening within this cohort of mutations. When we made a pool from bulk segregant pool and we did the bulk segregant fitness assay, we saw 2 really strong fitness effects. And those 2 genes were HSL7 and KEL1. You might remember KEL1 from a few slides ago. When we looked at how fitness segregates in this cross, we see a really skewed distribution where most of the segregants have a fitness effect comparable to the ancestor, and only a few segregants have a large fitness effect. So this distribution is one that we cannot explain based just upon generic diminishing returns epistasis. To get a better idea of what was going on, we took these 192 segregants, which up to this point were anonymous, and we genotyped them using low coverage whole genome sequencing. And so now we can split this up based upon the genotypes. And so when we break it down, we see that those that get just the KEL1 mutation are either neutral or perhaps slightly deleterious. Those that have the HSL7 mutation alone have a modest 3.5% fitness advantage. When you put KEL1 and HSL7 together, you get a real strong synergistic effect. So this is interesting because now we have a case of strong synergistic epistasis occurring at low frequency within these populations before this cohort of mutations swept to fixation. And so just to summarize, I told you very briefly about the previous work that we did looking at producing a very detailed picture of the dynamics of adaptation in experimentally evolved populations. How we're now trying to use tools, harness the power of yeast quantitative genetics to dissect fitness and epistasis in these crosses. Specifically, we're trying to look at how fitness segregates within a cross. We're using a bulk segregant fitness assay to very rapidly measure the background average fitness effects of mutations across lots of lineages. We're trying to develop a framework based on a paper that, like I said, Dan Weinrich wrote that will allow us to put these 2 data sets together in a way where we can make some good concrete objective statements about which populations must have idiosyncratic epistatic interactions in them. And in doing this, we identified one strong synergistic interaction between HSL7 and KEL1. What we'd like to do next is to really bring this full circle and start linking the emergence of epistasis in these populations to the dynamics of adaptation. And with that, I want to thank my lab. So most of this work was done by Sean, a postdoc, and Remy, a former undergrad. I had various technical help and funding, and thank you guys for hanging around on the last day.
Well, I have a question. So obviously you have these clones, as you say, groups of mutations, and when one of those with 5 comes along and wipes out the one with 3, there must have been something beneficial in the 3s that were lost. So an interesting thing to look at would be mixing those into the 5s in one of your big assays.
Yeah, so one of the things we're going to do next is start looking at epistasis, not crossing evolved to the ancestors, but crossing evolved strains to each other. So epistasis between populations. We expect to see a lot more there as well.
Okay, thanks.